Welcome to this video on the regulation of cell division. If you haven't got a handle on cell division, you probably want to get that first before we get into this one. Otherwise, it's probably not going to make a tremendous amount of sense for you. In this video, we're going to look at why cells need to control when they divide, particularly in multicellular organisms, the consequences of not doing that, and then we're also going to look at how cells control division, what they use in order to accomplish that process. In a multicellular organism, uncontrolled cell division is known as cancer. It is a group of diseases that are all due to some population of cells somewhere in the body that are dividing when they shouldn't be. This population of cells gives rise to a tumor, and so we can see a variety of tumors in the liver of this individual in this picture here. They are the white masses that are spread throughout the organ, and that's really where the disease comes from. Having these tumors growing inside of the organ disrupts the functions of the organs, and then can disrupt the homeostasis of the organism. Cancer can also, unfortunately, spread throughout the body. So cells from the tumor can break off and move throughout the body and can start to form tumors in other organs throughout the body. This is a situation known as metastasis, indicative of late-stage cancer, the kind of thing that's very, very difficult to treat. If you can catch cancer early, you have a much better chance of treating it than if you catch it later on in the progression of the disease. So it's really important to control when a cell divides. You do not want cells to divide except when they're supposed to. And so this diagram shows some of the different proteins that are involved in regulating the division of a typical human cell. And they all have different names here. You can see them like MEK and adenylate cyclase and all those sorts of things. And the other thing that you can see is that there are a variety of external controls, signals that are being sent to the cell and helping it understand if it needs to divide or not, as well as a variety of internal controls. Now, the specific nature of these controls is beyond our need to understand in this course, but we should understand that we have a variety of different signals that are working. If we're thinking about cell cycle controls internally, we can think about three major groups of genes that are involved in this process: prot-oncogenes, tumor suppressors, and DNA repair. Prot-oncogenes are any of the genes in our genome that stimulate cell division. They have a positive relationship with cell division. Those prot-oncogene proteins encourage cells to divide. Opposite in function to prot-oncogenes are tumor suppressors. Tumor suppressors have a negative role to play. They have a negative impact on cell division. And so if a tumor suppressor protein is present, it is going to make the cell less likely to divide. It will block cell division from occurring, maybe by blocking replication or blocking the mechanics of division, or whatever it is. That protein will prevent the cell from dividing. And so inside of a cell, in order to divide, you should have prot-oncogenes present saying divide, and you should not have tumor suppressor proteins present at that time so that there's no signal to stop. And so you need to have multiple things occur inside of the organism in order for cell division to occur abnormally. Specifically, you need to have mutations in prot-oncogenes that turn them into what are called oncogenes. The prefix onco refers to cancer. When prot-oncogenes are mutated and turned into oncogenes, they're going to overstimulate cell division. They're going to work when they shouldn't be working, sending the cell signals that it should divide when it really shouldn't be. Additionally, tumor suppressors are going to need to be mutated in order to become non-functional. So there is no signal to the cell that says do not divide when it should be dividing. And so in order for a cell to become cancerous, you need to have both of these things occur. You need to have oncogenes present, sending a signal of hyperdivision, saying divide, 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 divide. And you also need to have tumor suppressors not present, so that there are no signals to the cell that say do not divide when the oncogenes are telling the cell to divide. If you get both of these things to occur in multiple different prot-oncogenes and tumor suppressors in the genome, this is what needs to happen in order for a cell to become cancerous. Both of these things need to occur at the same time. The other major group of genes that are present that play a major role in controlling cell division are the DNA repair genes. Your DNA is constantly being mutated by chemicals and through cell division, through all these things, and you have a variety of different enzymes whose entire job is to go through your DNA and to fix the mutations that are occurring. In mutant situations, these genes become non-functional. And so as a result, your genome begins to accumulate mutations and does not fix them. The end result of this, of course, is that you wind up getting mutations in your prot-oncogenes and your tumor suppressors that make them much more likely to become non-functional and lead to the development of cancer. So while you absolutely do need mutations in your prot-oncogenes and your tumor suppressors simultaneously in the same cell in order to become cancerous, you don't need to have mutations to DNA repair genes to do the same thing, but really it just increases the likelihood of cancer. As we wrap up, let's talk a little bit about cancer treatment. This person is having chemotherapy, which is a typical way in which cancer is treated. Chemotherapy refers to a variety of drugs that are injected into somebody with cancer that preferentially destroy cells that are actively dividing. The idea being that the tumors in your body would be among the most actively dividing cells, and so they'd be the most likely to be destroyed by the chemotherapy agent. Different types of cancer will call for different types of chemotherapy agents depending upon the particular genetic makeup of the tumor and the progression of the disease, etc. Of course, there's all sorts of side effects that come along with chemotherapy due to the fact that the chemotherapy agent is killing cells that divide in your body, and many cells other than cancer cells divide in your body. Things like your follicles for producing hair, the lining of your stomach, etc. And so there's a variety of really unfortunate side effects that go along with a treatment like chemotherapy. Treatment like exposure to high energy radiation does the same thing, in that directing high energy radiation to the tumor will wind up destroying the genome of the tumor and can lead to the tumor no longer being able to divide and kill the tumor. It certainly doesn't always work, but it's a major approach. Certainly if the tumor is in an accessible place, surgery is an option. Doctors can go in and remove the tumor, but a lot of times tumors occur in regions of the body where it may not be possible to remove them through surgery, or the tumor may be too small for the surgeon to even see. There are a variety of newer approaches to cancer treatment that are individually tailored to a particular nature of a particular cancer. It's important to understand that while all cancer is uncontrolled cell division, every cancer that ever occurs is a unique cancer. It'll have a unique combination of particular mutations in ways that have affected those proteins involved in regulating cell division in particular ways. And by understanding the genetic profile of a particular tumor, that can help to tailor particular treatments for the specifics of the mutations that are present inside of that tumor. And that becomes much more common these days as our ability to understand the sequences of DNA in the genome have grown tremendously over the last few decades. Many of these individually tailored treatments still remain very experimental, but there are definitely more and more of them over time. Of course, the best thing to do to try to avoid cancer as much as possible is to modify your lifestyle so that you're much less likely to develop cancer. This is why, for instance, it's a bad idea to smoke cigarettes, as cigarettes greatly increase your likelihood of lung cancer. This is not to say that everybody who develops cancer is getting it because of bad lifestyles. In fact, most cancers develop as a result of random non-lifestyle choices. But still, by living lifestyles that lead you to avoid more common triggers for cancer, you're going to be less likely to develop cancer than you would be otherwise. The other major way that we get at cancer is through early detection. If we can detect signs and symptoms of cancers earlier on when they occur, it's much, much easier to treat them than later on once they've metastasized and spread throughout the body. As we move into check your learning here in this video, make sure that you can explain why control of cell division is necessary in living systems. Make sure that you can describe how cancer results from changes to cell cycle control proteins, and make sure that you can explain why cancer disrupts the functioning of a multicellular organism. If you can do those things, you're doing great. If not, that's okay too. Take a moment, pause the video, and write down any questions that you have so that you can get the answers that you need. Thanks again for watching. I really appreciate it. Have a good day.